everyone, my name is Mr. Motsapi from Tsongkota Secondary School here in PCM. Today's lesson will be talking about the basic agricultural genetics, and I will be breaking it down into the 4 subtopics: your genetic concepts and the law of inheritance, your variations, your patterns of inheritance, your selection and breeding. Those are the subtopics that we are going to cover for today's lesson. As we all know that basic agricultural genetics is a topic for paper 2, question 4, section B, it's 35 marks, question 4, and then the 15 marks will also come from section A, which covers 15 marks. So in total, this topic, it weighs 50 marks for paper 2. So let's take the terminology that is used in agricultural genetics, the genetic concepts. So we say that genetics is the scientific study of heredity. The scientific study of heredity, that is your genetics. So heredity is a concept that needs to be broken down on its own. So heredity is the transfer of characteristics from the parents to the offspring. The characteristics are moving from parents to the offspring. So in simple terms, we can say now, genetics is the study of how characteristics they move from parents to the offspring. So we want to study now how are characteristics, if you have got the black hair color and then you give birth to a child that also has the black hair color, how does that happen? So that is basically the concept of genetics. So those characteristics now, which can be passed on from parents to offsprings, we call them the hereditary traits. Those are your hereditary characteristics. These are the characteristics that can be passed on from the parents to the offsprings. Now we look at the concept of a gene. We say that a gene is a small section of the DNA that carries this hereditary information of a particular characteristic. So your gene is going to be found in the DNA, particularly in the chromosome. And we say that the location of that gene in the chromosome is what we call a locus. So a gene will be found in a chromosome, and the position of a gene in a chromosome is called a locus. Now, as much as we have a gene, this is what controls a characteristic. It's a gene. But a characteristic, you've got 2 alternatives. For instance, let's make it simple. We talk about the height. Height is our characteristic. Now, we talk of one characteristic which is controlled by one gene, but you can have tall or the height can be short. But we are speaking about one characteristic here, which is the height. But we have got 2 alternative forms of this height, which is tallness and shortness. So we are saying now we've got the alternative forms of the same characteristic. So these alternative forms of the same characteristics, we call them the what? The alleles. So the allele is the alternative form of the same gene. So in other words, one gene contains 2 alleles, which are the alternative forms. A gene controls the characteristic. In this case, our characteristic is the height. But the height have got 2 alternative forms, which is the tallness or the shortness. Your organism can be either tall or can be either short. So those alternative forms of the same gene are called the alleles. Now, your alleles, we've got 2 different types of alleles. You have what you call a dominant allele and the recessive allele. A dominant allele is the one that is going to mask the effect of the recessive allele. In other words, in the presence of a dominant allele, the recessive allele is not going to appear in the organism. So for instance, when you talk about the height, if we say tallness is dominant over shortness, it means now that the dominant allele is going to mask the effect of a recessive allele. When we have got a tall organism and a short organism, you mate them to produce the offspring. The offspring will appear to be tall, even though there's a genetic that comes from the short organism, because the shortness is recessive. So a dominant allele will mask the effect of a recessive allele. And your recessive allele, which is another type of allele, this one is the one that is marked by the dominant allele. So in other words, okay, we'll come back to this concept again. Let's move on and talk about what we call the phenotype. The phenotype is the physical appearance of an individual. So what you see is a phenotype. So this is the outward appearance, the physical appearance. When you say you see a black cow, that black is the phenotype. When you say you've got a tall plant, that tallness is the phenotype. So the physical or the outward appearance of an organism. That's what we call a phenotype. And then we also have what the concept of a genotype. This is the genetic composition of an individual, of an organism. So in other words, your genotype consists of the allele that comes from the father or the paternal side and the allele that comes from the maternal side. So it's a genetic composition. We have got this allele and this allele. So there are two alleles. One comes from the paternal side. The other one comes from the maternal side. These two alleles now, they form a genetic composition of an organism. We call it a what? A genotype. But a genotype cannot be seen by the eyes. We cannot see the genotype. But very, very important, the genotype, this is the one that determines the physical appearance. So the genotype, in simple terms, determines your phenotype. But your phenotype is the physical appearance. Now let's come back. The dominant allele is the one that masks the effect of a recessive allele. It therefore means the dominant allele is the one that will always determine the physical appearance. Now, when the dominant allele is present, it's the one that will determine. But a recessive allele will be masked by the dominant allele. But very, very important, it's very key this point. A recessive allele, sometimes it can determine the phenotype, but only under the homozygous conditions. But a dominant allele, under homozygous conditions and under your heterozygous conditions, it will always determine the phenotype. But a recessive allele will only determine the phenotype in the absence of a dominant allele. So that's what we call homozygous. Homozygous means having the same alleles for a particular characteristics. So, for instance, if we say here, the organism is tall, and this particular organism is having two alleles, capital letter T and capital letter T. So we can refer to that organism as homozygous. Homozygous means having the same alleles, that is homozygous. And then we have what we call heterozygous. This one means different alleles for a particular characteristic. So if we have capital letter T and small letter t, this is your genotype. But in this genetic composition, we have different alleles. The other one is dominant, the other one is recessive. That one is having different or alternating alleles. That one is called heterozygous. So hetero means different. That is the meaning of hetero, different. And homo means the same. That is the meaning of homo. Homo means the same, hetero means different. So we look at now the type of inheritance. Now there are two types of inheritance, your monohybrid cross and the dihybrid cross. So very, very simple. Monohybrid cross, this is a reproduction between two parents. Very, very important, between two parents. But it involves the inheritance of only one characteristic. That's a monohybrid cross. And then your monohybrid cross will have two steps. The first step and the second step. The first step is called the parental cross, and the second step is your F1 cross. Your parents are always homozygous. A homozygous organism is referred to as a purebred. And your F1 organisms are all the same. They are all the same, very, very important. And they are always heterozygous. A heterozygous organism is called a hybrid. So those two concepts are very important. Now they will give rise to the F2 generation. In terms of F2 generation, the genotypic ratio will always appear to be 1:2:1. And the phenotypic ratio, now the physical appearance, will always appear to be 3:1 in a monohybrid cross. So we move now and look at the dihybrid cross. We move and look at the dihybrid cross. So this one is also a reproduction between two parents, but now we are concerned with the inheritance of two characteristics. Remember, in a monohybrid cross, we are looking at the inheritance of only one, but now we are looking at the inheritance of two genetic characteristics. So the theory is still the same, divided into two steps. Step number 1 is the parental cross. Step number 2 is the F1 cross. Your parents are also homozygous. They are purebreds. A purebred is a homozygous organism. So very, very important, these concepts, because when we ask questions in agriculture, sometimes we're going to say a purebred black cow was crossed with a purebred red bull. So you need to understand a purebred is a homozygous organism and a hybrid is a heterozygous organism. So same thing, all the F1 organisms, your F1 represents the first generation. They are always the same. They are all the same, and they are always heterozygous. Then they give birth to the second generation. So when you look at your F2 generation, the phenotypic ratio there is going to be 9:3:3:1. That is the phenotypic ratio of the dihybrid cross. It's just the same as a monohybrid. The difference is that here we are looking at two characteristics. Now, let's quickly do this one now so that we can close this chapter of the genetic crosses, and then we can easily explain the laws of inheritance. So, this is an activity that I've just chosen for us to quickly understand, which I took from Agric Science paper 2, November 2023. In pigs, red hair color is dominant over white color. A white sow, which is the female pig, your sow is a female pig, it was crossed with, or it was mated with a red boar. This one is a male pig, a red boar. And all the piglets of the F1 generation were red. All the piglets, these are the offspring of the pigs, they were all red. Use a Punnett square method. To determine the genotype of the F1 generation. Now, very, very easy. Already you know from the content that we have covered that your parents are purebreds. And then your F1 generation, they are all the same and they are heterozygous. So you just draw your Punnett square. Just draw your Punnett square. This is your Punnett square. So here you have the gametes of your parents. These are the gametes. And then you are told that you have, so here, remember we have got the gametes, but the simplest way for us, for learners, is to just draw this line. This line shows that there is a reproduction that is taking place. So it simple says there's a fertilization. Fertilization, we know that is the fusion of gametes. So we have got the gametes here. Your parents are purebreds. Parents are purebreds. So use a Punnett square. Already we have got the answer. You are told that all the piglets of the F1 generation are red. Now, you have the first piglet, the second piglet. They are always the same and they are all heterozygous. So this is your Punnett square. Very, very important. So you just get that 4 marks from drawing up your Punnett square. But very, very important, here you have got the gametes of the parents. So sometimes the question will say, you draw a Punnett square to determine the genotypes of the F2 generation. Then you will use the gametes of the F1 generation to get the F2 generation. So, okay, so now we look at these laws, Mendel's law of inheritance. Now, the first law, Mendel's first law, is called the law of segregation. The law of segregation is known as Mendel's first law. This is Mendel's first law. And then the law of independent assortment is known as Mendel's second law. Very, very important. Now, let's look at first law. The first law says each characteristic or every characteristic is controlled by a pair of alleles. It's controlled by two factors. These two factors are alleles. It's a pair of alleles. So there are two factors, which is a pair of alleles. These alleles, they separate randomly during the process of meiosis so that each gamete can receive only one allele. So we already know that if we say we have this one, is the red, these alleles during meiosis, they separate so that each gamete can receive only one factor or allele. That is the law of segregation. But very, very important, the law of segregation, or Mendel's first law of inheritance, it only applies when we talk of the what? The monohybrid crosses or monohybrid inheritance. Then we move to the second law of Mendel, which is the law of independent assortment. Now, the law of independent assortment, this one, it states that the pairs of alleles, they assort themselves independently of each other after the process of meiosis. So this one applies during the dihybrid inheritance, which is now the inheritance of two characteristics. So we are simply saying here, if you have a round and it's a round, let's talk about the seeds. The round seed as well as, let's say it's a round seed and it's a black seed. Round and black seed. It's a round seed and it's a black seed. Maybe it was crossed with a wrinkled seed and a white seed. You can see these are your parents. They are purebreds. In other words, they have the same alleles. Now, the law of independent assortment, it simply says that during the process of meiosis, a pair of alleles, they sort themselves independently. Remember, they will separate randomly. So you're going to have this capital letter R for color assorting itself with this one. Another one with that one. So they are independently. This pair is independent of this pair. Same applies that side. This pair is independent from the other pair. So that is the law of independent assortment. Very, very important. This one, it applies when we talk of the dihybrid inheritance. So first law is when we talk about monohybrid crosses. Second law is when we talk about the dihybrid crosses. So that concludes now the genetic concepts as well as the laws of Mendel's law of inheritance. So now we are going to move and then we talk about the part 2 of our lesson for today. Part 2 is the patterns of inheritance as well as variations. We're going to look at the patterns of inheritance as well as variation. Now, let's go and talk about patterns of inheritance and variation. The patterns of inheritance as well as variation. So, we first look at the concept of the dominance. Dominance. Now we have got 3 types of dominance: complete dominance, incomplete, and the co-dominance. Very, very important. Complete dominance, one allele is dominant and the other allele is recessive. So this one is what Mendel actually used in his studies when he was using the pea plants. He used where it's just a dominant and a recessive allele. But now, other geneticists, they discovered later on that there are other variants or variations in terms of the phenotypes now that can be explained beyond the concept of Mendel. That's why now we talk of the patterns of inheritance. So we look at the concept of the incomplete dominance as well as the co-dominance. Incomplete dominance, no allele is dominant over the other. And when you have these two parents coming together now, they produce the third intermediate phenotype. So we can simply say you have a red flower. This is your red flower, was crossed with a white flower. This is your white flower. Remember, no allele is dominant over the other. So we're not going to use small letters. A small letter represents a recessive allele. So when these two now come together, they produce a pink flower. So a pink flower, it's the intermediate of red and white. So that one is called the incomplete dominance. Your organism is not red, it's not white, it's the intermediate. So that one is the incomplete dominance. And then we move and look at what we call the co-dominance. Co-dominance. Now, when we look at the co-dominance, when we are looking at the co-dominance, this one, both alleles are equally dominant. Both alleles are equally dominant. So it's different from the incomplete dominance where no allele is dominant. So here we are going to say you have a red cow that was crossed with the white bull. Now they produce a roan offspring. So both red and white will be equally expressed or displayed in the phenotype of the offspring. That one is called dominance. A dominant allele will always appear in the phenotype. So if we say both of them are dominant, both of them will be displayed in the phenotype. So that one is called the co-dominance. And then we continue with these patterns of inheritance. We continue with patterns of inheritance. We look at the multiple alleles. Very, very simple. But now we are changing now from what Mendel have actually discovered. One characteristic here is controlled by more than two alleles. So remember, Mendel told us that a characteristic or a gene have got two alternative forms. But now we are saying one gene can have more than two alternative forms. That one is called multiple alleles. Prepotency, this is the ability of one parent to pass on its genetic characteristic to the offspring. So you have two parents, but one parent has the more ability to pass on its characteristic. For instance, if you say you have a red bull that was mated with the white cow, a red bull mated with the white cow, and all the offsprings of the first generation are red. It therefore means that your red bull has the more ability to pass on its characteristic to the offspring. Because remember, if phenotype is what we see, when we look at this offspring, we are just going to see the red offspring instead of seeing the white. So that one is called the prepotence. And then we move and look at what we call atavism. Atavism, this is the reappearance of the ancestral characteristic after it has been absent for a long time or for generations. So that's what we call atavism. For instance, if you say in the Angus cattle, you have got the red Angus, but somewhere, somehow now, there is a reappearance of a black Angus cattle. So now that means we talk of the atavism, the reappearance of the ancestral characteristics after it has been absent for quite some time. And then we also look at the polygenic inheritance. Polygenic inheritance. Poly means many. Very, very important. Poly means many. So here now we have many genes. But these genes that are many, they control one characteristic. So that one is called the polygenic inheritance. You usually see it in characteristics such as your milk production, your height. So those characteristics are controlled by the polygenic inheritance, the size of your plant sometimes. So those characteristics are controlled by polygenic inheritance. Then we move, and then as we continue with the patterns of inheritance, now we have what we call the epistasis as well as the sex-linked characteristics. Epistasis, this one now, is a pattern of inheritance in which the effect of one gene is controlled by another gene. So there are two genes that are interacting here. One gene is the gene that controls the characteristic. Then we have another gene which controls the effect of this gene. So this other gene now which controls the effect of one gene is called an epistatic gene. So we usually see it in characteristics such as the color coat in the horses, the coat or the color coat or the feathers in the chickens. So that's where now you see the epistasis. One gene controls the effect of another gene. So that is the pattern of inheritance, which is called epistasis. We have come to end of our lesson. You can leave your questions on the comments. Don't forget to leave the name of your school so we give shout out to your school. Please share, recommend, and subscribe to our YouTube channel. Thank you for watching. See you on the next lesson. Thank you very much.